Hi students, welcome back to Ligase Academy, your educational catalyst. Today we are going to discuss about DNA replication in eukaryotes. We have already discussed about prokaryotic DNA replication in the previous videos. Before explaining DNA replication in eukaryotes, let me just remind the points we have already discussed. DNA replication is the process of producing two identical replicas from one original DNA molecule, right? The fundamental mechanism of eukaryotic replication is same as prokaryotic DNA replication, but there are some variations. The replication in eukaryotes are more complex because of some reasons. The reasons are eukaryotic genomes are quite complex. They are considerably larger than bacterial DNA. They are organized into complex nucleoprotein structures, that is chromatin. Essential features of DNA replications are the same in prokaryotes and eukaryotes. Similarities of prokaryotes and eukaryotic replications include formation of replication fork, semi-conservative replication, movement of replication fork bidirectional, primer synthesis, Okazaki fragment synthesis in lagging strand, primer removal, gap bridging between newly synthesized DNA fragments. The difference between prokaryotic and eukaryotic replications include the overall process of eukaryotic replication is a bit more complex. Important differences are due to larger size of eukaryotic DNA compared to prokaryotic DNA and also distinct package of eukaryotic DNA in the term of chromatin. And also they have slower rate of fork movement in eukaryotes. For DNA to become available to DNA polymerase, nucleotide must disassemble. This step slows the rate of fork movement. So these are the similarities and dissimilarities between prokaryotic as well as eukaryotic replication. So in this video, we are discussing about initiation stage of eukaryotic DNA replication. So eukaryotic DNA replication occurs at a particular stage of cell cycle, that is S phase of cell cycle, that is synthesis phase. As we all know, the eukaryotes have a linear chromosome and the proteins involved in replications are initiator protein is ORC, that is origin recognition complex. Then helicase is MCM complex, that is mini chromosome maintenance complex. Then helicase ladder is CDC6-CDT1. Primase is Pol alpha, that is primase. Then single strand maintenance protein is RPA, that is replication protein A. Sliding clamp is PCNA, that is proliferating cell nuclear antigen. Then clamp ladder is RFC, that is replication factor C. DNA polymerase enzymes are DNA pole delta for lagging strand, DNA pole epsilon for leading strand. Then for primer removal, FEN1, that is flap endonuclease 1, then DNA 2, then helicase 2. And ligase is DNA ligase 1. So these are the proteins involved in DNA replication in eukaryotes. As in the case of prokaryotic replication, eukaryotic replication also contains three stages, namely initiation, elongation, and termination. In this in this section, we are dealing with initiation. So, eukaryotic genome contains multiple replicons. So, the origin is ARS, that is autonomously replicating sequence. It have 11 base pairs conserved sequence called as A element, that is ACS, that is ARS consensus sequence. In the image, there is B2, B1, A and C, and where the orange color 11 base pair is the sequence of A element, and the ORC is the initiator protein. So this initiator protein will bind to the ARS, that is autonomously replicating sequence. The major two events occurring in initiation are replicator selection or origin licensing and origin firing. So first is replicator selection or origin licensing. It involved recruitment of MCM complex. MCM complex is helicase to ARS. ARS is origin by ORC, that is initiator. So remember that all replicators are not selected at a single time. So when ORC, that is initiator proteins, binds to ARS, that is in origin, it recruits MCM complex into ARS. So MCM complex is helicase. So helicase is loaded by with the help of helicase loader. Helicase loader is CD C6 CDT1. So whenever ORC is binds to ARS, then MCM complex is loaded into the ARS with the help of CDC6 CDT1, that is helicase loader. ORC, that is initiator protein, is a heterohexamer A plus protein, and helicase loading is done by CDC6 CDT1. MCM complex is helicase. It is a heterohexamer and it is MCM 2 to 7 complex. It is an inactive helicase. So, two such helicases are loaded onto double stranded DNA in opposite orientation. MCM bound AR is called as pre-RC, that is pre-replicative complex. It is a licensed origin. So, origin licensing occurs all stages of cell cycle except S phase. So predominantly it occurs in G1 or late M phase. The second stage in initiation is origin firing. It occurs in S phase of cell cycle, and S phase specific cyclins activate two serine threonine kinases such as CDK and DDK. CDK is cyclin dependent kinase and DDK is DBFO dependent kinase. DDK has two subunits such as catalytic subunit as CDC7 and stimulatory subunit as DBFO. So DDK phosphorylate MCM complex, whereas CDK phosphorylate SLD2, SLD3 and ORC. So phosphorylated ORC is targeted for degradation. So origin licensing will not be possible in S phase. The phosphorylated SLD2 and SLD3 associate with DBP11 and form a scaffold structure, and it is recruited to the MCM complex. So SLD3 recruits CDC45, that is cell division cyclin 45, and SLD2 recruit pole epsilon and GINS complex. GINS complex is a heterotetramer that contains 4 subunits, and a multi-protein complex of MCM, GINS, and CDC45 called CMG complex forms. It is a holohelicase and functionally active. They start unwinding the DNA in opposite direction to create a replication fork. Additionally, pole delta and pole alpha is recruited into the DNA, and RPA will coat the unbound single-stranded regions of the DNA. Pole alpha is the primase in eukaryotic DNA replication. It is a DNA dependent DNA polymerase and RNA polymerase. They have no 3' 5' exonuclease activity. They do not have proofreading activity. So they has 4 subunit, that is a large subunit with DNA polymerase activity, a small subunit with RNA polymerase activity, and 2 small subunit, that is accessory subunits. So pole alpha synthesizes short RNA primer about 7 to 10 nucleotide long, and it is further extended by 30 nucleotides by its own DNA polymerase activity. So first it will produce some short RNA primer of 7 to 10 nucleotide with its RNA polymerase activity, and further it is extended by 30 nucleotides by its own DNA polymerase activity. So the resulting PT junction, that is primer template junction, is recognized by RFC which is the clamp loader, and that load PCNA is the clamp. So PCNA is bound by pole epsilon in leading strand and pole delta in lagging strand. So during initiation, there is a polymerase switching event in which primase is replaced by pole alpha, which is later replaced by pole epsilon or pole delta. So these switching events are mediated by RPA. So that's all about it. That's all about initiation stages in eukaryotic replication. Hope you all understood about what we discussed today. If you have any doubts or queries regarding the topic, please let us know through the comment section. If you like this video, please like, share, and subscribe our YouTube channel like His Academy, your educational catalyst. Stay tuned for the next video. Thank you.